Assalamualaikum and good morning to everyone. Thank you, Dennis, for the introduction, and also thank you to everyone who attending this NGS webinar. So today's topic will be presenting about Amplicon sequence analysis. Title is sample to data discovery the diversity of nature's microbial population through Amplicon sequence analysis. Right. Yeah. Please stay until the end of the session because we have some quiz and there's a gift to be won. Also, there will be a special announcement regarding the Amplicon sequence service and by Apical. So make sure to stay tuned until the end of the session, right? And if you have any question related to this Amplicon sequence analysis or related to our service, kindly type your question in the chat box. Please write in your question, your name, and your email, right? Because at the end of the session, we will select a few of the questions to be answered. And because due to the limited of time, so some of this question will be answered. The question will be right back to you. We will be right back to you by email. All right. Okay. So let's start with our presentation. So this is my presentation overview for today. So we'll start talking about microbes and metagenomic, and then we look into one of the application in metagenome, which is Amplicon sequence analysis, and then we look into the service that offered by Apical Scientific and then Berhad, and finally we look into the demo report, right? So we look what are the offer, what kind of the result that we deliver to all our customers. Right. So as introduction, so what is microbes? So microbe is, as you all know, microbe is a microscopic organism comprised of either a single cell, unicellular, cell cluster, or multicellular. It can be found everywhere. It plays a very significant role in the environment. So you can find them inside sea, water, inside soil. In fact, you also can find them in the animal guts, right? So microbes can be either a good or either a bad bacteria. So for example, microbes are a vital component of a fertile soil. So it contains billions of cells in the soil. Estimated just in a single gram of soil, there are billions of bacteria alone. Another example is like in human body, microbes make up the human microbiota, include the essential gut microflora. In our stomach contain billions of microbes, which play roles in the food digestion. So any changes in the gut flora can affect the human health. So this is a report. The studies have shown that there's in example in the animal study shown that any changes in the microbe profile in the stomach can contribute to their health. So one example they show in the mice. So any changes in the microbe profile contribute to the obesity. Right. So so for thousands of years, human has been exploiting microbes for human benefits. For example, 1,000 years ago, in Egypt, it has been reported that the microbes has been used for wine production. Today, with the advance of technology, microbe has been used for many things. For example, like from food to health, up to a large industry like energy production. Right. So like enzyme is very crucial for food production, like ketchup, like soy sauce, um, like fermentation. For all these things are required enzyme for usage. Uh, Another example like antibiotics as for drugs against a bacterial infection. These are just few examples of microbe usage, and there are many more. All right. Uh, and 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 for your information, so these benefits for the microbe are came from the cultured microbes. Uh, However, most unfortunately, it has been shown that it's actually that most of these micro environment are actually unculturable. It's reported that less than 1% less than 1% of these microbe can be cultured on the plate agar, and all this have been all this has we have been focusing on this 1% microbes for all this potential. So if you imagine that if you can tap this microbe potential from the unculturable microbe, so you can see the potential is endless. Therefore, this is a need to use a different approach to study microbes from the in the environment, right? So that's where this metagenomic approach comes in. So what is metagenomic? So metagenomic is a study of microorganism. So the keyword here is recovered directly. So it's a study of microorganism recovered directly from environmental or clinical samples, and later studied using by a functional gene screening or sequence analysis. So the main keyword here is recovered directly. So metagenomics keep the culturing process. So this enable us to look into the potential that contains the contained in the unculturable microbes. This approach, metagenomic, has been started in 1980s. However, during that time, um, the metagenomic only focusing on the functional based screening, that which you have to clone and then you have to express, and then you have to screen the your interest, the enzyme of the interest. There's also some groups that working on sequencing this the microbe genetic materials, but during around that time in 1980s to 2000, the Sanger sequencing, it's running the sequencing using a Sanger platform is very expensive. Not only that, you need to have trained technical person, and it's very laborious, from starting from getting the sample up to clone them and then sequencing. It take years to get the result to came out. So unfortunately, that time metagenomic is not that popular because it's not really accessible to everyone. Right. Everything changed when the next generation sequencing came in, which is in around 2006. So it revolutionized how we look into these metagenomic studies. The reason is because the NGS allow the sample to be sequenced without need to be cloned. In other words, that if you extract the samples from the environment, um, and then you just can proceed with library preparation, and you can directly sequence the sample. And not to mention the cost, if you calculate based on per GB, cost per GB from NGS and Sanger. NGS is very, very cheap. So that is that's when this NGS really revolutionized the metagenomic studies, and now we can see a lot of lab now work and move into this metagenomic study using the NGS platform. Right. So talking about metagenomic approach, now we focus on the NGS era. So we're talking about the NGS. We're talking about the sequencing. So the functional screening we are not covering here. So we just focus on the sequencing using the next gen sequencing. So basically, metagenomic can be there are two type of application here. There are two types of approach. The first one is Amplicon sequence analysis over here. And another one is the metagenomic sequencing, is shotgun metagenomic sequencing. So what's the difference between the amplicon sequence analysis is that for amplicon sequence analysis, we just target a gene marker. So this gene marker must be present across your organism, target organism. Right, for example, like 16S or REG-A gene or ITS, right? So it depends on your gene target. So you amplify this gene target and you sequence, and using the bioinformatic analysis, you study this the information that contains in this gene target. On the other hand, for shotgun metagenomic sequencing, you don't amplify or you don't target any of the gene marker. What you do is you just sequence the whole genomic content here, and then after the sequencing part, doing using the bioinformatic tools, you try to assemble back this metagenome, right? Try to reconstruct back the metagenome, and from there we can do a study from the gene that is contained inside this metagenome, assembled metagenome. So from there we can study like the functional information. So and also if the what's the organism diversity. So yeah, so you can also look into the diversity and also the functional information. So there are two big difference here. So the amplicon sequence analysis, you only focus on the gene marker, while the shotgun metagenomic, you sequence the whole genome. Talking about the cost, if you look into the amplicon sequence analysis, because you only focus on the gene target, the cost is cheaper compared to shotgun metagenomic because we have to sequence a lot. The more coverage we have, the better. Because as you know, the metagenome consists of mix of microbes. So that's the reason you need to have a high amount of coverage to to have a very good representative of the metagenome. So the cost is a factor as well. Mm. But in terms of information that can be extracted from amplicon sequence analysis, you only provide you like the diversity information, like what are the microbes that's present, and also how much are they inside the environment. There's no or limited function information present in the amplicon sequence analysis because we don't really look into the functional gene, right? So the function information inside if user amplicon sequencing will be unavailable or very limited. Unlike metagenomic sequencing, because you sequence the whole metagenome, so you can do the gene annotation, and then you have access to the gene information inside the DNA metagenome. So you can also access the gene function information in the using the shotgun metagenomic. Right, so basically that's a simple a brief comparison between the amplicon sequence analysis and shotgun metagenomics. There are a few more, but I'll just highlight the most. They are the one of the most different between the amplicon sequence analysis and shotgun metagenomic. Right. So for today's discussion, so I will just focus on the amplicon sequence analysis. And for shotgun metagenomic sequencing, hopefully we have another session to talk about that, right? Or if you have any question related to shotgun metagenomic, you can always email to us. We can always help you to look into the inquiry. All right. So amplicon sequence analysis. So basically for amplicon sequence analysis, the workflow for amplicon sequencing can be divided into three phases or three steps here. So the first step will be the sample preparation. So the sample prep means that from the mix of microbial community, so you need to extract the DNA metagenome. So you need to ensure the quantity and the quality is good. And then you can proceed to the second stage or second step. So in the second step, you will do the PCR, lab reconstruction, and sequencing, right? And the outcome for the sequencing will be a lot of sequence reads, right? And all the sequence reads need to be processed in the third stage, which is in the bioinformatic analysis, where they'll summarize all these reads into like a tabular format, like where you can get something like this, like sequence read blue color, and then you can get the abundance information. And from using matching to the database, you're able to know what are these sequence matched to what species. Right. So this is based on a basic, simple workflow. If you have one sample, uh, what if you have multiple samples? Like for example, like here you have microbial community A, and you also have the mixed microbial community B. For example, you have a sample isolate from different soil. Let's say you have microbes from paddy soil. You want to compare against microbes that is contained inside oil palm soil. For example, you can use the same approach. So have to extract the DNA metagenome, do a PCR, library construction and sequencing, you get the data, and from the BI analysis, you can do a comparative analysis as shown here. So we can do a compare the abundance between the environment A and environment B. So from here, we can know what are the microbes that enrich in one environment and what are the other microbes that are kind of like low. abundance in another environment. Not only that, we can also do some other analysis like statistic analysis, alpha, beta, which I will explain. I mean, which I will cover a little bit in the towards the end of the slide. Okay, right. So come back to this question. So who should use this amplicon sequence analysis approach? So this is just some idea. So like for example, who those who are in the livestock and farming industry, they can use this approach to study the soil health. For example, like they saw in the paddy field, one part of the land the production is very low, and another one is very they can give good yield. So perhaps you want to know what's the microbe that contain in this plot of land. So perhaps this microbe might contribute to the production of the paddy. Other example like want to study the feedstock, so you want to know what are the feed that is benefit to your animal, to the animal, to the livestock. So yeah, so study what are the good probiotic that can be given to the livestock. All right, in start in for wildlife, you can also study the animal diet. For example, those like tiger, we can collect the stool from the tiger, and then we can do the amplicon sequence analysis. From there, we can get information what this animal eat. So yeah, so this can give some idea, understanding. So how this animal, the wildlife, the health, and what they eat in the environment. Apart from that, in the health sector, we can also look into the gut health, because the gut health equals to the healthy lifestyle. So we can collect the stool, and then we can study what's the microbe proportion inside the stool. So from there we can study whether this person has a good gut health or not. Then perhaps we can change the diet or do something to improve the gut health. In the food industry, for example, you can it's very important want to know whether are we producing the consistent product. So for example, that in cheese, for example, so you want to make sure that we always give produce a consistent product. So we need to make sure that the starting culture are the correct one. So we can use this approach to study to ensure that everything are consistent. Also for university and local research institute, like usual, you can use to do a fundamental study of microbes and to study its diversity. All right, so this is just example. Who can use this approach? I believe the list will be endless. So if you want to know whether this approach suitable to you, you can also write to us and ask. We can have a discussion about your project, and then we can assist you from there. Right. So now let's look into the service offered by Apical Scientific Center Berhad. So we offer amplicon sequence analysis. All right. So to send the sample to us, we can look into the requirement. So there are a few things that we need to look into. The first thing is the DNA requirement, the sample requirement. And the next, we look into the gene target. Gene target is very important. It depends, it will determine what are the microbe groups that you are looking at, and also the sequencing and also the read amount for the generated for the process. And finally, we look into the demo report. You can look into how our result looks like. Okay. Right, so let's look into the first one, the sample details. So for sample requirement, we accept genomic DNA, accepted genomic DNA. So we only need around 200 nanograms, so it's not a huge amount. So 200 nanogram of DNA, which is we need to have them in a volume more than 20 microliter, in concentration more than 10 nanogram per microliter, right? And purity must be good. OD60 to OD260 must be around 1.8 and 2.0, and must ensure there's no degradation or no contamination. Um, we also offer DNA extraction service. If let's say you have a difficulties on getting a good DNA yield, we do have this service. You can send the material to us. We have experience extracting the DNA from various part of sample, from soil, stool, water. So we have our protocol to do DNA extraction service. So yes, you can also send sample material to us. We can do the DNA extraction for you. Uh, right. So that's about the sample requirement. Now let's look into the gene target. All right. So the gene target is very important. For example, if you want to look into the prokaryote, so you should look into the CDS variable region. If you are studying on fungi, then you should look into the ITS1 or ITS2. For small eukaryote, you can use the 18S V region. Right. We also accept other type of gene target, for example, CO1, tRNL. As long the amplicon size range must be around 350 base pair to 500 base pair. So this is very important. The size range must be 350 base pair and 500 base pair. Later, I will explain why the size is only limit to 500 base pair. All right. So as I emphasized just earlier, so choice of target region is very important. Because if you use a different target region, then your target organism, target microbe will be different. Right. From our experience, we call it 3S1W. 3S stands for soil, sludge, stool, and water. Seems to be very good using CTS V3, V4. For other type of environment, it will be advisable for you to refer to for publication. See what this author used to use for as a target region. That will be the best, right? So you can share with us the target region. We can design the primer and we can do the PCR for you. Okay. Right. So this is just now I mentioned about why it must be around the region must be around 350 base pair to 500 base pair, right? The reason is because the Illumina platform unable to sequence a large fragment size is only limit the maximum size to 500 base pair, right? Yeah. So if you see if you can see this picture, this is the diagram of a CTNS. So the CTNS contains of multiple variable region from V1 to V6, right? So using a different primer, we can use to target this variable region. So one of the most widely used variable region is a V3, V4. So which generate size about 400 to 500 base pair. So this is just one example. Some other groups might use a V4. It depends on the group target organism they are looking at. So for example, the different of the gene target will produce a different microbes, will target different type of microbes. For example, if you can see, this is published by Roche et al. In 2019. So what they did is they have a mock sample. Mock sample means that they cultured and prepared a mix of microbes at a known proportion. So they have prepared this mock sample, and then using a different variable region, they try to see they attempt to the amplicon sequences using a different type of variable region. So V1, this one V1, V2, this one V3, V4. So you can see the profile of microbes are different. Some are good to capture this group, and another one, this type of V3, V4 might be suitable for different type of groups. So it is important that for you to select the variable region, it's wise to refer to a publication so that you will look at the correct information. Okay, but from our experience for 3S1W, soil, sludge, and stool plus water, V3V4 performs very well. So that's what I would advise for any one of you working with this environment, soil, sludge, and stool plus water. We can advise you to go for V3V4. Right. Um, right. So now let's look into the sequencing and rate amount. So for Apical, samples are processed and sequenced using on MySeq platform. So we have a MySeq platform in our lab, right? So we sequence in house. So all the samples will be sent for sequencing on MySeq platform after the sample QC and PCR QC are passed. And we for the sequencing output, uh, so the sequencing format will be the 300 base pair per M, And for every project, for every sample, we target around a minimum of 200,000 raw reads per sample. So this will be per sample. So if let's say you have 10 samples, then each sample will get 200,000 raw reads or equals to 100,000 raw text per sample. All right. So yeah, so Amplicon, our service, Apical Scientific Sample Hut, offer a one-stop solution starting from a sample prep, PCR, library construction, sequencing up to bioinformatics. So we have a master platform in-house. So we have experience delivered hundreds of Amplicon sequencing project. We have a very fast TAT, which is 6 week, around 6 week after QC pass, and we also offer technical support if you require any of the support after we deliver the result. Right. Right. So for the result deliverables, so we can look into we provide few type of result which later we can share the report, the demo report to you guys. Okay. So we provide result on taxa bar plot, we provide result heat map, distant matrix, alpha, beta, diversity, pie crust. Pie crust is only applicable for 16x only. So if let's say your samples looking into the 18S or looking into the ITS1, ITS2, unfortunately, we cannot provide the PyCrust because it depends on the PyCrust database. So as of now, the PyCrust database only works well for the CTS target region. We have provide also the Corona chart, UPGMA, and also the Venn diagram. Okay, all right. So let's look into the demo report. Uh, I can share you the link after the end of this session. I will share you the link to our demo report. You can download this demo report to your PC and you can look into how is our demo report looks like. All right, so let's jump into this. Okay, allow me to open my file. Yeah. Okay, all right. So this is our demo report. So our result are being provided as in a HTML format. All right. So all the analysis are been linked into the tab file. So no worries. This just for your information, this file is actually is offline. So all the result are be linked in the folder. So it's not an online file. So it means that even without internet, you still can view all the result. Okay. Right. So all because all the file are linked into the folders that been provided to you. Okay, so you can see there's a few tabs here, so which you can click, right? So all the link here represent the analysis that have been provided to the customer, right? So I will just briefly go through with this result, okay? And later after this talk, you can download and then you can try out yourself. And you have any question, we can always assist you. Just write to us, then we can assist you from there. All right. So in the first page, we have about introduction. Then we have talk about the approach that we use for our service. We use a data to pipeline to analyze the data, and you can see here there's a flow, a simple workflow for the data to pipeline, starting from the FASTQ file, pass to the quality assessment where we check the data whether we have enough as amount of data as promised to the customer. And once it's passed, then we can put it inside the data to pipeline to process it, and it will came out as a ESV table. And for the ESV table, you can do many things like you can do a diagram, you can generate a reflection curve, alpha, beta, diversity analysis, statistic, and so on. And all the list of softwares and also the reference are being listed here, which is useful for publication. And we have a rate statistics tab. So we mentioned about the rate statistic from the raw data up to the process rates. And from the process reads, we will analyze that and generate the ESV table, which you can see over here. So for this ESV table, you can have the sequence ID here. And then next to it, we have the abundance, right? Some information about this demo report. So this is a demo report which contains 9 sample. So 9 sample and 3 groups. 3 groups, 9 sample means that each group contains there are 3 groups, 3 sample per groups, right? So in total they have 9 samples. So we have a group A, group B, and group C, right? So we have this information. So you can see the numbers here represent the abundance information. And towards the end of this table, you can have the taxonomy information. You can also access the sequence here by clicking this link over here. They will access to the sequence, process sequence. All right, so from the MySet data, we process them, merge them, and from there, they will summarize the sequence here. And this sequence are being used to generate the abundance and also the taxonomy information, right? So in case that you want to know further about this particular sequence, for example, so you can always like click this sequence, you get the link, click this link, which open this file, and you can just copy this ID over here, and you can do a search, Ctrl FB, then you can find the sequence that match to the ESV number one, right? So you can do like blast or further down analysis as you want. Okay, you can also have this file in Excel file just by clicking this download ESV table. So they will provide the same table in the Excel format, because some of you prefer to look this in the Excel format, right? uh, this is just a table. We also provide the information, the same information in view several type of figure or diagram. For example, you provide that in the bar plot. So this is our bar plot over here. Okay, so if you click this link, by default it show the bar plot at the genus composition, but we also provide the bar chart at different type of taxonomy level. So you can just click here, then you can get the bar chart at different taxonomy level. One thing about our bar chart is there's some features here which is very interesting. For example, you can, if you move your mouse over the line over here, they will explain the information. So you don't have to mark the color and then try to find what are the information contains here, right? So it will be easier for our customer to look into the result by moving the mouse over the region, right? And not only that, the we can also do a selection. For example, like like if you like to do a selection, you will target a certain group of organism. For example, let's select a microbe here. For example, let's say you are interested to look into the enterobacter, so you can just double click here. So they will straight away highlight the just that selected organism. So they will make the search much easier, right? You can have this selection, and you want to return back to original, you can just double click and then it will turn back to the original state. Okay, all right. Send the bottom, you have the information um, based on a top 10 composition. For example, if you are, the information are too complex, perhaps you would like to focus on the top 10 first, then you can look into the top 10 graph bar chart here. They also there's also the bar chart for just grouping information. So this is bar chart for group A, group B, group C, means they have consolidate all the replicates into one, and will provide the bar chart based on the grouping information. Right. So this is the abundance based on the text, based on the bar chart. We also provide the result based on the heat map. Basically it's the same thing. Right, let's look at the heat map genus HTML. Right, so basically they show the same information except that this is based on the heat map format. All right, so yeah, if you prefer to present in the heat map, so you can use this information. Apart from that, we also provide the result based on the corona chart. So this is a corona chart result. So for one corona chart here represent one sample. Since that you have 9 samples, so basically we have 9 chart. So if you click here, you can navigate across the samples. Okay, and you using the corona chart, you can do a samples like grouping selection. You can go you can select group of microbes. Like for example, you want to focus on the protobacteria, for example. So you can just click this, and then you click one more time. They will expand the chart, and then focusing only the protobacteria. And if you navigate across all the sample chroma chart, all the chroma chart will be set to the same group. So easy for you to do a comparison across all samples like this. Okay. And if you want to save this picture as your publication, you can just right click and then you can copy or save image, and you can use that for publication. All right. Yeah. Apart from that, you also have the alpha and beta diversity. So we have a few indices. So we have the alpha. The description about the alpha indices are being listed here. All right, and then at the bottom we have the reflection curve. So we provide the reflection curve to to explain whether uh, are we sequence enough to capture all the microbe diversity in the environment. So we can see here all the samples, all the line here reach the plateau. So means that we have covered our sequencing have covered most of the microbes inside an environment. Right. Apart from there, we can also have the beta diversity or ordination. So they can be used to study the microdiversity across different different environments. So we can use a beta diversity. So there are few indices provided like CCA, MDS, NMDS, PCOA or RDA. We also provide a 3D PCOA plot, right? So which we can move according to the which suitable to present your data, right? So that's about the ordination. um we also provide apart from that, we have the um like the comparison analysis like diffusion abundance. So this is only for diffusion abundance. Here is only applicable if you have more than two groups, because we need to do a comparison analysis. So to compare, you need to have at least two groups, and each group we need to have at least three samples, because there's a statistic to be applied. So for statistic analysis to be applied, you need to have at least a minimum of three samples. So for any project that require diffusion abundance, you need to have a minimum of two groups. Each group must have at least three samples. Then we can provide the different abundance information. So for diffusion abundance analysis, there are a few type of algorithm analysis that we provide to you, like DESSEC, LEFC, Metastat, and so on. So yeah, you can see the result like examples here. For example, if you click the DESSEC, let's say in the genus, and if let's say on the PNG here, you click the picture, uh, well, you can, this for example, the first one is a comparison between the group B and the group C, right? So the group B and the group C. So the result will explain what are the microbe groups that have been enriched, and the statistic will provide you whether this information are significant or not. So that is why you need to have a replicates at least a minimum of three groups, so that we can have a statistic to support the information. And for example, what this data set tells that in group C, the G lactobacillus are higher in group C compared to group B, while for Levilactobacillus and also Providentia are higher in group B compared to group C. So this is one of the things that you can do. Yeah, we also provide the result. This is based on the some examples, all right? So we also provide, you also have the result in Excel format. So the table, um, all right? So the result are provided in Excel. So we can open this file in Excel and allow to fix a bit. All right, so this is the whole list. Just now we just look into some few top 5 sample of the result of the test 2, B versus C. So this is the whole list in the Excel file. So from here we can see what are the taxa that have been enriched in which group, and what are the difference between one group to another. All right, so we can see here, for example, this group, the fold change between one group to another is about minus 23, and then it's very significant, and then it's enriched in group C, so it means that it's higher in group C. All right, so these are just some example. There are a few types of analysis. This is only a dissect. There are a few more, but unfortunately we don't have time to go through the rest. But you can, once you can try out using our link. If you have any question, you can come back to us for discuss if you want to know further. Apart from that, we also have a Venn diagram. So there's a usual, I mean, I think you guys are familiar with Venn diagram, right? So we can, before we start with the analysis, we provide you with the BI form, so we can list out how we want to compare your samples. Can be based on sample or based on grouping. For example, this is one example based on grouping. So the demo report, here we list out the Venn diagram based on the sample group A versus group B and group C. So the Venn diagram here will explain what are the samples, what are the microbes that are unique to one to another. Okay, all right. And lastly, we have want to explain about the pie crust. All right, so this pie crust, as I explained in the slide, is only applicable if you are working with 16S, right? Because the pie crust database covered CTS database very well. So we are unable to provide pie crust result in different type of target region like ITS1, ITS2, and so on. Microstome, what does we give is using the microbe that is present inside the environment. They can predict what are the gene information, what are the gene content, and then from there we can, by compiling all the microbes that present environment, we can plot. They can Pycrast can plot a graph of functional gene in functional information. Bear in mind, this is just a gene prediction based on the taxonomy, based on the microbe that present in the environment. So it's not a direct gene prediction. It's not like you sequence the gene. It's just that based on the microbes. From there, they estimate what are the enzyme or what are the gene that might be present inside these microbes. For example, if you're working with from fermentation from those microbe might contain genes that can be survive in the aerobic environment. So we expect that all those genes might be present inside this pie crust analysis. So maybe I can just show one example here because we have uh, limited of time, right? So this is just example from the pie crust analysis. So we can open, let's say, the based on the EC result. All right, so this is what we can get from the pie crust analysis. The pie crust analysis will provide the EC and also the description of the this enzyme information, and then then the next we have the abundance information uh, of res of respective enzyme here across all samples. From here we can estimate what are the genes that might be present in the environment. Right, so that's just something that we can look into using the pie crust result. All right, so that's the last part of our demo report, which I can explain today. Yeah, the last part will be about our company and also our partner, which help assist us on the bioinformatic analysis. All right, so that's the last part of my presentation. So pass back to Dennis for next session. We have a QA session or what?